What if we could edit cancer out of a person's body? What if we had the power to find the exact genetic typo that fuels a disease like pancreatic cancer and simply delete it? That's the profound promise of CRISPR, and it's rapidly moving from the realm of science fiction into the reality of our labs. Imagine your DNA as a vast, intricate library of books containing the complete instructions for building and operating your body. Every cell holds a copy of this library. Cancer, at its core, is a disease born from errors in these instructions, typos in the genetic text that cause cells to grow uncontrollably. For decades, our approach to fighting cancer has been like trying to get rid of a bad chapter by burning the entire library with chemotherapy or blasting it with radiation. These methods can be effective, but they're blunt instruments, causing immense collateral damage to the rest of the body. But now, we're on the cusp of a revolution. We're developing a tool that doesn't just attack the cancer, but precisely rewrites its source code. Think of CRISPR as a pair of microscopic, programmable scissors, a biological word processor with a find and replace function for our very own DNA. These molecular scissors can be programmed to navigate the entire genetic library, find a specific, faulty sentence like the mutated gene driving a tumor and make a precise cut, effectively editing the error out of existence. This isn't just a new drug, it's a fundamentally new paradigm, a next-generation approach to medicine that treats disease at its most fundamental level. In the complex and aggressive fight against pancreatic cancer, this programmable technology is being aimed at two primary battlefronts, each with its own strategy for rewriting the rules of the disease. The first, and most audacious, is a direct attack on the tumor cells themselves. Pancreatic cancer is notoriously driven by a handful of well-known genetic mutations, with one villain standing out above the rest, the KRAS gene. In over 90% of pancreatic tumors, a mutated form of KRAS acts like a stuck gas pedal, constantly telling the cancer cells to divide and spread. The visionary goal of CRISPR research is to program those molecular scissors to enter pancreatic cancer cells, hunt down that specific, mutated KRAS gene, and make a precise cut. By snipping this faulty gene, we could disable the cancer's primary engine. It's the equivalent of finding the master switch in the cancer's command center and flipping it to off. This approach aims to stop the cancer not just by killing the cells, but by removing the very instruction that makes them cancerous in the first place. The elegance of this strategy is its precision. It promises a future where we can disarm the tumor from within, leaving healthy cells untouched. The second battlefront, and the one closer to becoming a clinical reality, involves supercharging our own natural defenses. Your immune system is a powerful, vigilant force, constantly patrolling for threats, including rogue cancer cells. However, aggressive cancers like pancreatic cancer are masters of disguise and suppression. They create a fortress around themselves, a dense, fibrous microenvironment that shields them from immune attack and sends out signals that effectively put immune cells to sleep. This is where CRISPR becomes a powerful tool for re-engineering our own soldiers. The strategy, known as CAR T-cell therapy, is a marvel of personalized medicine. Doctors start by taking a sample of a patient's own T-cells, the elite special forces of the immune system. Outside the body, in a lab, they use CRISPR's precise editing capabilities to rewrite the T-cell's genetic code. They are engineered to produce a new, synthetic receptor on their surface, called a chimeric antigen receptor, or CAR. This new receptor acts like a highly specialized GPS and homing missile, programmed to recognize and lock onto a specific protein found on the surface of the patient's pancreatic cancer cells. Once these supercharged, reprogrammed hunter cells are multiplied by the millions, they are infused back into the patient's bloodstream. They are now a living, programmable drug, a personalized army designed with one mission: to seek and destroy the patient's unique cancer. CRISPR allows scientists to make these edits with unprecedented accuracy, building a better, more resilient, and more lethal T-cell, turning the patient's own body into the ultimate weapon against the disease. So, with this incredible technology, is CRISPR the real cure for pancreatic cancer that we've all been waiting for? Let's be honest and clear, it is not a cure today. The leap from a brilliant concept in the lab to a standard treatment in the clinic is a monumental one, filled with massive hurdles. One of the biggest challenges is delivery. How do you get these microscopic scissors safely and efficiently to the right place? the dense, fortified pancreatic tumor, while bypassing the rest of the body. It's like trying to mail a letter to a specific address inside a heavily guarded fortress. We also have to ensure absolute precision. While CRISPR is remarkably accurate, the risk of off-target edits, accidental cuts in the wrong part of the DNA, is a serious concern that scientists are working tirelessly to eliminate. Finally, the pancreatic tumor microenvironment itself remains a formidable barrier, a complex ecosystem that actively works to thwart any attack, whether from chemotherapy or engineered immune cells. We are still in the foundational stages of this research, building the very first tools and learning how to use them. But even with these challenges, it is impossible to overstate the profound, long-term hope that CRISPR represents. While it's early, this technology is more than just another potential therapy. It represents a fundamental new way to think about curing cancer. For the first time, we are not just fighting the symptoms of a genetic disease, we are developing the ability to correct the disease at its source, by rewriting its very genetic code. This is about building the tools for the next generation of therapies. A future where medicine is not just about managing disease, but about precisely correcting the biological blueprint. It's a shift from waging a war of attrition against cancer to performing precise, genetic surgery. This is the pace of innovation that will ultimately unlock the cures of tomorrow. The path from a laboratory breakthrough to a patient's bedside is long, and it is paved with painstaking research, rigorous trials, and immense dedication. But every single breakthrough, every cure we have today, started exactly like this, as a vision, a bold idea that challenged the limits of what was thought possible. Here at Pancreatic Cancer Warriors, we believe in the power of that vision. We are tracking these developments closely, translating the science, and sharing the hope that comes from knowing that the brightest minds in the world are working to rewrite the future of this disease. The revolution is coming, one edit at a time.